protein, look, these are only proteins are being synthesized. These are not infectious. These are harmless. But because SARS-CoV-2 viral proteins are being formed, our body recognize it as a foreign, and we can synthesize antibodies against these viral vaccines. So this is what it gets injected into the volunteer, and then RNA coding for the antigen starts synthesizing these antigens, that is basically the spike proteins, and then our immune system cells started recognizing it as a foreign and start immune response and immune memory. This is how the procedure for the development of the RNA vaccine technology works, friends. So, could you able to see a small video?
Yes, sir. Yes.
Yes, sir, we are able to see.
Yes, sir. It can be.
I'll stop here. Could you able to see Dr. Meena?
Yes, sir, we were able.
Yes, sir. It
Yes,
is
sir.
visible. It is visible and it's
Fantastic. So this is a sequence, and they identify the spike protein. So it's a vaccine candidate, right? And this mRNA, they inserted this spike protein into the mRNA and they synthesized it, right? This mRNA strand. So they put it in the RNA vaccine. So basically lipozyme mediated RNA vaccine. And once you put that into the lymph node, basically thymus gland, then our immune cells in the lymph node, it started expressing the viral spike protein onto the surface. Then our immune body, the antibodies started detecting it as a foreign, and our body start making antigens. So this is in nutshell how the RNA vaccines work. So we really don't have to expose ourselves to the viruses. All we need is the protein, right? So you can see that it is originally isolated from infected patient. Then the sequencing, work has been done in China. Then the vaccine developed in the US, and then right now it is in the clinical trials. So this is how the journey of the Moderna vaccine is all about. So the drug discovery is exciting field, and how about ourselves? We were talking about the vaccines. All we need is got two approaches. The first approach is the classical approach, which is screening for bioactivities. So you can see that which is trial and error or tinkering based approach. So then we keep on trying different kinds of bioactivity like done in China. Then the vaccine developed in the US, and then right now it is in the clinical trials. It's just like medicinal plant chemistry. So we extract the medicinal plant and test it against the various bioactivity like anti-cancer, antimicrobial, antiviral, like that. This is a virus, so it's an antiviral effect of the various plant extract. So our cell line, which is usually used for the SARS-CoV-2, is HEK293 and VAR-E6 cell line. So that is what the SARS-CoV-2 can grow, and then they test against suppression of the SARS-CoV-2. Now, which plant extract to use? We have 8.7 million species of eukaryote. And now you take the extraction. There are four kinds usually chemists use: aqueous extract, alcohol extract, phenol extract, and chloroform extract. So 8.7 million multiplied by 4 is the total number of combinations, 34 million. So it's not easy to test against each and every plant species for for bioactivity screening. It takes long time and it's massively expensive, and rampant red tape bureaucracy prevent export of the medicinal plants in most of the countries. For example, Kenya, for example, Kenya has got a lot of plant diversity, but Kenyan government completely forbids export of any medicinal plant from the Kenyan territory because they are really afraid that rich countries will they will make use, so they will make multi-million dollars out of it, and they will not pay anything to the Kenya. And the situation is exactly the same here in India as well. The governments will not allow you to export the live medicinal plant because we are all afraid that other countries can make use of it without paying us anything. Could you please mute? All of you, please mute. I can hear some disturbance in the background. So this is what perhaps you might know. This is the first line. When the coronavirus came a few months back in the mainstream, and this is the first, the drug which have been used widely. This is nothing but hydrochloroquine. This has been isolated from cinchona bark. The bark of the cinchona plant in Amazonian rainforest. It has been used for thousands of years by these tribes in Peru and Chile, and these tribes are known as Quechua tribes. Quechua tribal medicine. That
Hello.
is what the ethnobiology or ethnobotany is all about, isn't it? But
Are you able
unfortunately,
to
the
hear me?
hydrochloroquine, I mean, it's very effective for anti-malarial drug, but for the coronavirus, we know that it doesn't work that well. Nowadays nobody is actually administering the hydrochloroquine because it gives us more harm than good, right? So I have covered everything in the curiosity. I do have a YouTube channel. You can search my name, Felix Bast, and I do have a program called Curiosity on every month. I cover the top science stories of the month, previous month, and also opportunities for students and researchers. So please search my name and subscribe to my channel. So in the COVID-19, this a plant extract based therapy. One of the promising candidate is the seaweed extract, which outperforms remdesivir. Remdesivir is the most commonly used drug now against COVID-19, though it is not commonly available here in India. We have no really effective drug, but remdesivir is used in Europe as well as in Japan and US. So even it beats remdesivir, the seaweed extract. So seaweed is nothing but macroalgae. Marine macroalgae is called seaweed. So the researchers have isolated a complex sulfated polysaccharide, the fucoidines from the kelp, that is brown seaweed. The scientific name is Saccharina japonica. So this sulfated polysaccharide. So polysaccharide is nothing but sugar. Sulfated means there is sulfur containing sugar, and this is known as fucoidine because the name fuco means fucus. So fucales is the family of the saccharina, so that is why the name fucoidine. But friends, mind that tons of treatments show promise in cell cultures but fail for various reasons in the clinic. One example would be hydrochloroquine. You know, so until you are put through the human trials, this is just hype and hypothesis. So human trials is really important. I've covered again in the curiosity. Now coming to the combinatorial chemistry approach to find the drug. This is the easiest way, and this is now least expensive and now more prominent. Everybody is after this combinatorial chemistry thing. So how does this work? So this is basically structure-based drug design, or also known as rational drug design. So basically, one team will look at the structure of the coronavirus in very detail, extremely high resolution. They actually do this one in this proteomics setup. So it's basically, as you can see, this is multi-TOF and X-ray crystallography. So once you do this X-ray crystallography, the three-dimensional structure of this viral spike protein or any features of the virus becomes very evident, and they upload the structure to the protein database. The structure is available. So this work is done by proteomics team. So now this is what the protein data bank is. PDB or CSB. PDB is a very famous site, again managed by the NCBI. So you can search 6VXX. This is the accession number for our COVID-19. This is a SARS-CoV-2 spike glycoprotein closed state. So of course it has got two states, closed and open. So it has got two accession numbers. So once you know the spike protein structure, so for a sake of simplicity, let this be the structure of the spike protein, right? The pink, as you can see on the screen, is the spike protein structure. Now this is a receptor. We need to find the ligand that can block this spike protein from binding to our cell, like mucosal cell, like our respiratory canals. So we have to find a ligand. So basically ligand could be most often plant extract. So how do you find it? So we have got libraries of different plant extracts. So for example, Interbioscreen is a very famous library, or you can go for Analyticon Discovery or Albany Molecular Research Inc. or Biosocia Pharmaceutical. I mean, a lot of tons of different natural product structural libraries are available. So this one example would be Interbioscreen. We have got 4 potential ligands of plant extracts: plant 1, plant 2, plant 3, and plant 4. So basically this work will be done by structural biologist who is working on the plant. So these two are interrelated. One team will work only on the receptor structure elucidation. Another team will work only on generating the structure, and the third team will do the docking. So if this is our ligand and this is our receptor, that is spike protein receptor of the coronavirus, which plant extract would bind it perfectly? Any guesses from the audience? You can unmute and you can speak on. Say your name. So we have purple extract, we have green extract, we have yellow extract, and we have blue extract. So which extract binds perfectly?
Blue.
Yes. What is your name?
Shekhar
Yes.
Chaudhary.
Yes, Shekhar Chaudhary. Correct. It's blue. Yeah, blue looks like identical, right? It can actually bind it. But of course, we can just by looking. This one is extremely simplified, but in reality, the protein structure is 3-dimensional. So it's really difficult to make a guesswork. So we have to test the thermodynamics equation. So this is the Gibbs free energy equation. Delta G is equal to delta H minus T delta S. So delta G is basically the free energy. So if you minimize this free energy, this is the aim to achieve the good ligand substrate docking. Ligand and substrate, or receptor and ligand. So the docking, what we have to look is delta G has to be very less. So free energy has to be less so that it binds very nicely. So if you look at this software like Glide, then it will generate the delta G with different receptor and ligand. Here you see the ligand is all same. I mean the receptor is all same, which is the receptor is spike protein. But ligands we have 4 different plant extract. This purple extract we have got delta G is 88, while green one it's 7, and this yellow one it is 124. And while this one, this blue one, it's -2. So delta G could be minus as well. So the lowest delta G here is this, -2 kilojoules per mole is a unit of this delta G here. And the worst fit is the highest one, delta G, right? This is the worst one. So by looking at it, we know it. So of course you can use any of these free molecular docking software. So I'm telling you this. These work you can do at home, sitting in your home with a small computer. You can simply download any of these software and you can search in the YouTube for various training of how to use any of these software. For example, ProDoc. ProDoc is a free software released by Cornell University, which is based on Monte Carlo method plus energy minimization. So it's a Bayesian thermodynamic optimization algorithm. So you should know how to use this software. These are all free softwares. And of course, we have got the spike protein. Of course, the PDB has got the 3-dimensional structure of it. And we have Interbioscreen and other natural products library. So we just have to dock it. And whichever things docking very nicely, you can even write a paper out of it without doing any wet lab work. So that is the beauty of it. But of course, you should learn how to use this software. So you can do it if you spend a bit time with YouTube and downloading the software. So yeah, instead of simply wasting your time in social media, you can do this to publish some quality work. And of course, there's a few papers on that lines as well. Ribavirin, remdesivir, against SARS-CoV-2 RNA-dependent RNA polymerase, that is RDRP, a molecular docking study. So this study has revealed the utility of remdesivir because remdesivir can block the expression of this enzyme RNA-dependent RNA polymerase. Remember, our virus is single-stranded RNA virus. So it needs this crucial enzyme, RNA-dependent RNA polymerase, that is RDRP. So if you can prevent this enzyme from expressing, then the virus cannot perform its replication, right? So that is what this remdesivir does. So that is after this publication of this paper, the remdesivir started extensive use, especially by Gilead Pharmaceutical. They did this study, and yeah, of course, you can do this way as well. Huh? This is another study. Computational screening of antagonist against SARS-CoV-2, COVID-19, coronavirus by molecular docking. So you can see a lot of this kind of papers coming up these days. So you can do this molecular docking and to find the antagonist. So antagonists prevent expression, while agonist, another term agonist, that actually improve the expression level. So these two things are different. And this is another very interesting paper which we have covered in Curiosity channel as well. Neutralizing nanobodies bind SARS-CoV-2 spike RBD and block the interaction with ACE2. ACE2 is a receptor where the SARS-CoV-2 bind to human epithelial cell. So this one, usually we know our human antibody is IgG and IgM. So the antibody is pretty big. It cannot effectively bind to the grooves of this SARS-CoV-2 spike protein. So that is why people are now looking for llama, not Dalai Lama, but llama is it's basically a camel family, camel-like animal in the South America. It lives in Peru, Chile, and those Brazil and all those areas in South America. So this could llama antibodies treat viruses like COVID-19? Yes, bioinformatics people are working on those lines as well. So it could be the weapon against COVID-19. We have another approach, that is RNAi approach, that is RNA interference approach to find the drug. So it's basically RNA interference or RNA silencing. As you know, RNA is very important. Messenger RNA, it actually gets translated in the cytosol to form the protein. So if this RNA can somehow get prevented from the translation, so here the translation is prevented by antisense DNA oligonucleotide that can bind to the RNA. So that is what this RNA silencing is all about. So for that work, the Nobel Prize for Physiology of Medicine in 2006 were awarded to Andrew Fire and Craig Mello. These two scientists got this award. So as you know, the two approach for this one is, one approach is basically the siRNA, synthetic small interfering RNA based approach, while the other approach is miRNA, which is more or less the natural one, microRNA approach. So any of these approach you can use. The crucial enzyme involved is dicer, and RISC protein, and DOSA is involved. So basically what it does is in both the pathways is it degrades. It binds and it degrades and prevents from the expression of this protein. So you can do that. Now the point is that where to target, which gene to stop from expressing. So we can look either at the spike protein of this SARS-CoV-2, that basically is a spike protein. Of course, there is an mRNA, and that mRNA can be silenced in the virus. Or we can also look at these receptors in the host cell. Basically, this is our cell, human cell. We have ACE2, or we also have got TMPRSS2, and we also have ADAM17, SERM, CALM, molecule. All these molecules are basically, as you can see, these are androgens, estrogens, SERM, and androgens. So all these come antagonists. So all these are a potential target that can be silenced by that. So as you can see, one very interesting paper has come up in Frontiers, Prospects for RNAi therapy for COVID-19. So if you look at the targets, as I told you, in host or receptor, in the host, there is our own cell. You can silence expression of ACE2 receptors. So basically the beta blocker drug, if you have somebody who has heart failure or heart patients at home, so they will be taking this beta blocker. So basically ACE2 receptor, if you silence expression of ACE2 receptor by RNAi, Issue is that the blood pressure could drastically come down, so that could be dangerous to the host. So that is why it is not really a recommended practice. It has to be. Uh, even people are looking for ACE2 inhibitors for SARS-CoV-2 therapy. So another one, a better approach, I would say, is on silencing the gene expression in the pathogen, that is SARS-CoV-2 coronavirus. So silencing the expression of S glycoprotein, E, M, and N protein. All these proteins are involved in the RNA, the expression of spike proteins in the coronavirus thing. And now finally on the evolutionary origins of SARS-CoV-2, I really don't have time. I want to get some comments from you as well. I have covered this in my YouTube channel. You can have a look at that. And let me just go. Yeah, all these things are there in my channel, and also many important, because I can see that many of you are students watching this video. Around 30 students are here, and the students also covered so many time management and personal productivity tips, and and the personal productivity 2-part series, academic writing. Several of these videos are there in my channel, so please have a look. I also have this curiosity, which is science show. It's a monthly science show, only 20 minutes, but I cover almost 20 to 30 most important and significant findings of the previous month, and also the opportunities, including the PhD and postdoc and grants and upcoming sky watching events, and observations. All those things I cover in the curiosity. Nanni is Malayalam word for thank you for watching, and this is my curiosity, my YouTube channel. That is, let me read out for you. bit.ly/bastf, BASTF. I read again, bit.ly/BASTF, Bastf as one word. And thank you so much. And now I would like to hear some of your comments, or any questions if you have.
Thank you, sir.
Yes,
Enough
ma'am.
phone.
Yes.
Thank you, sir. Such a nice presentation, and we are extremely sorry for initial technical errors. But your presentation is now was going on the YouTube live, and I hope everybody assessing it. And if left something or information, you already shared some links, and some that's a good link, and we are really going through their YLS and curiosity. Several videos are there, and they are awesome, and they're very good for students and anybody. So that presentation is very great and very valuable for all our participants. So now we are ready for taking few questions if there is in chat box. Sir, if you allow us to take some questions.
Yeah, go ahead.
Oh, thank you, sir. Just few seconds.
There were 107 participants from YouTube also, and one of them asked, sir, about immune boosters' role against COVID, sir.
Okay, so this one is I have covered this in my channel quite often. Immune boosters doesn't work. So immune boosting itself is a dangerous practice, because if you boost your immune system, that will result in autoimmune diseases like rheumatoid arthritis, and many of these diseases, for even diabetes. Diabetes mellitus is insipidus, for example, is a autoimmune disease. So the boosting the immunity is not a good idea, and also it is impossible to boost your immune system. We have got only one option, that is therapeutic option is only one, that is basically vaccine. If you get vaccine, then of course the body produces antibodies against the whatever the pathogen is. Another option to get the immunity is to expose yourself to the COVID-19. So if it is not so life-threatening disease, one example would be chickenpox. So chickenpox is hardly life-threatening as a child, but it could be dangerous as an adult. But as a child, if you get chicken pox, not a big deal. So even people, deliberately expose a child to the chicken pox to get the immunity. Yes, that practice is common in developing countries. So there is also known as variolation. But in the case of COVID-19, no, it is not a good idea to expose yourself to COVID-19 to get immunity, because it's a life-threatening disease, and that is why many people they don't really understand the concept of the herd immunity. So herd immunity is basically indirect immunity, because everybody else got infected and recovered from it, and because of that you are kind of protected. But again, herd immunity doesn't work for COVID-19. So to get it worked, you have to get infected and then recover from it. So chance of successful recovery itself is not that easy in the COVID-19, because the COVID-19 successfully recovered. You might have seen that successfully recovered. The person is successfully recovered. But friends, there is nothing called successful recovery. If you ever got infected with COVID-19, your heart muscles are irrevocably damaged, and you have got micro clots all around your body. So you are, highly significantly prone to develop the heart diseases in the future. Lifelong threatening is there, and you would be in need of the lifelong blood thinners, and we are only coming to know recently. And one of my closest friend recently, he passed away. So he got the COVID-19. He was very young and fit. He is a national level boxer, and he is our yoga instructor. He's a religiously performing yoga every single day, and he conducted several yoga sessions, and he's also believing in these immune boosters. And friends, we lost him. You know how he got COVID-19, and then he was in the hospital. He successfully recovered from it. He tested negative and he is back home. Then on one night he had a silent attack, and he died. So this is a recurring theme with COVID-19. The disease is really dangerous, and because of this immune boosting myth, people consume these herbal immune boosters, and it's really dangerous, because they think that they have a false sense of protection, and because they think that they're protected, they venture out in public without wearing mask and without observing any physical distancing, and then they get it. That is actually the hard reality. And even nothing, immune boosters doesn't work at all. In this India, we have got so many products claiming to be immune boosters. Even bread, modern bread, and Born is another bread company. They say immune booster. They just add a little bit of turmeric and they claim that it is immune booster. Even sari is coming, immune boosting sari. They add some little bit of the turmeric and other herbs, and they claim that it is immune booster. Even mattress, friend. Arihant is a mattress company based in Mumbai, and they put one whole sheet of advertisement that our mattress is immune boosters, because they added a little bit of turmeric in their mattress. And this country has lots of immune boosters, and unfortunately India is one of the worst affected in the whole world. And countries like Japan or Western Europe, or even New Zealand, for example, don't have any of these herbal immune boosters. They are much well off. So the immune boosting is a myth, and please don't fall into this immune boosting myth. Yeah.
Thank you, sir. I think the answer we got for this. There are some more comments in the chat box, sir, if you can take one or two. One, Soma Sundaram says that, and 2-3 from YouTube link also, that when will the vaccination reach the common man in India, according to you, sir?
Vaccination, I have a great hope in this Oxford vaccine, Chadox and COV2, as well as this Moderna. Moderna is basically the mRNA vaccine. So they are actually under clinical trials, human clinical trial. Laborious clinical trials are really important. Unfortunately, Chadox vaccine have to put on hold, because if one person have any complication, even if he gets some other disease, so that is exactly what has happened. The person got some other disease during the vaccine trials. For example, a person might already be having some heart disease and he got a
I
heart
have a
attack, for example.
question.
So we, it's not easy to. No, let me complete this question. So it's not easy to rule out
Uh,
the causative factors for all these things. So first you have to rule out the cause of this one, then only the vaccine can. So in my opinion, it might take any time between next few months, maybe 3 to 6 or 7 months for the first drug candidate to hit the market. We also have one vaccine called Sputnik by the Russia, but unfortunately, nobody believes in it. It's just a political propaganda, just like the Russian propagandist regimes are doing. Yeah.
Thank you, sir. I think we'll take a question from Professor Rama Rao from Northeast Shillong University. Sir? Rao, sir, you can ask the question now, please, sir. I think we have a connection problem.
Um,
I think we have some connection problem there.
Okay. Then I can pick up a question here. Sangram Keshri Panda is asking, why remdesivir is allowed for treatment of COVID-19 as it has no positive effect towards the disease? In this way, who is responsible for this? Is there any penalty for the company? So these are actually, even you might know that Donald Trump, when he got admitted, he got this remdesivir. So as of now, we really have not a single drug. So experimental drug can be used given the patient consciously allow for it, and or the patient's family signs the agreement to be tested for this experimental drug. So there is no problem in it. If you're okay to be tested with experimental drug, so it is ethical. It's a human ethical practice. So I don't think any unethical thing that the company behind the remdesivir has done, because they get PIC, prior informed consent, or rather FPIC, free prior informed consent. So once you get it, why not? As of now, we have nothing to lose or nothing to gain. So that is the reason that we are actually getting, and it's not just for COVID-19, even for several of these cancers. We don't have any the drug, so people can take the experimental drug and contribute in that drug development. Yeah. So there is another question here. Soma Sundaram is asking, sir, there are many vaccines under trial, and so when the commercialization of the vaccine comes, and how it's certified worldwide. So worldwide certification doesn't exist. There is the country. It's up to the country to approve or not to approve. For example, Sputnik seemingly approved in the Russia, but everybody is now questioning the Russian development of that. There is no human clinical trial, and they are not releasing anything to the public. So probably it's just a propaganda. It's just a political game. And the question is that there are many vaccines under trial, and so when commercialization of the vaccine comes, so we have no idea. So the trials, there are three kinds of trial. The first trial, the clinical trial, is a very small scale. Second one is basically the human beings to see the adverse effect, and the third one is a human beings at large scale, thousands of human volunteers it will test. So even after getting this third stage of the human clinical trial, only it will. So when means no one knows. It might never come also. Who knows? Like for example, HIV. We have pumped in billions of US dollar for the development of the HIV vaccine, but till date we don't have a vaccine against HIV. It's also an mRNA virus, right? Just like coronavirus. So we might never get vaccine also. That might also be a reality. So we should come in terms with it. So, I don't believe that optimistic, being hopelessly optimism is a good option. Thinking only looking the positive side, being hopeful that vaccine will come. No, I think it's better option is think vaccine will never come and come in terms with the reality and live it. So proactive rather than reactive, right? It's always better to take adequate precautions. Yeah.
Yes, sir, we agree with you that we have to be very cautious. And presently, mask is the only vaccine, is the correct way to say things.
Yeah, we already have a vaccine, so wearing proper mask, mask wearing is a good vaccine against COVID-19 to get it. So by the way, I have seen many people are wearing mask incorrectly. Just check it out in my YouTube videos. The touching in the front part of the vaccine completely invalidates the wearing of mask. And whenever you want to take out the mask, only touch the loops, and you should actually put inside out and put inside the Ziploc bag. And whenever you touch the mask, you should clean with the soap water. So all these are very basic, but nobody does it. And I think that is one main reason why COVID-19 is transmitted. And the situation is really worse. So we really have to be very careful. Yeah. I think it's time. I already have another meeting 9:30. So thanks a lot for today.
Uh,
So I
Thank you,
enjoyed
sir.
this session.
thank you,
Thank you,
sir.
sir. Thank you, sir.
Thank you, Dr. Felix, sir. And it's such a great presentation and lots of questions our participant want to ask about COVID-19, and but it's a lesser lack of time, sir. As you wish. So thank you very much, sir.
Yeah, we can have another session if you like. Yeah. Yeah.
Yes, sir. And
Thank you.
that will be for a privilege for us if you agree for the next time, and that time we will be
Yeah,
more
sure.
proper and more prepared for that technical webinar and practices lots of ready to say. So thank you very much, sir. Be with us.
Thank you. Bye bye.
Bye, sir. Good night, sir.
Thank you, and that was a very good session. And we are getting ready for our second session of Professor N.S. Shekhawat, sir. And I think Ashok Patel is with us. He will introduce us, give us introduction to Dr. Shekhawat, and then over to you, Ashok. Na? Ashok, please unmute your mic first.
Okay. Is it audible?
Yes. Yes.
Okay. Thank you very much, ma'am. A very good morning to all the participants, eminent scientists, faculty members, delegates, research scholars, and the students present in this national webinar on recent advances in biotechnology. I, Dr. Ashok Kumar Patel, Assistant Professor, Department of Botany, Jai Narain University, will be moderating this session. I again welcome you all on behalf of the Department of Botany, Jai Narain University, Jodhpur, and I thank you all for spending your valuable time and being here with us today. I assure all of you that it would definitely be a knowledgeable and memorable experience for all of us in the present situation of pandemic, as we are facing a very unexpected and unprecedented situation and going online to stay connected. Now, I would like to introduce the next speaker, Professor N.S. Shekhawat, and I feel very much privileged and pleased to introduce Professor N.S. Shekhawat, who is a great botanist, biotechnologist, and an eminent scientist. Once again. Okay. Is it visible?
Yes.
Okay. The slide is visible or not?
It is visible. Make it full screen if you can. Present the slide in a full screen.
I think it's not visible to me. Okay. Dr. N.S. Shekhawat, who is retired as a professor of botany from Center of Advanced Study, Department of Botany, after a dedicated tenure of 36 years of teaching and research at Narain Vyas University, Jodhpur. During this period, he served as head of the department, in charge of Biotechnology Unit, DBT Network Project, and coordinator of UGC SAP, COSIST, and DST FIST programs. He had served as Director, Internal Quality Assurance Cell, University Science Instrumentation Center, and Educational Multimedia Research Center of this university. He has been postdoctoral research associate at Department of Biology, University of Yale, New Haven, USA, with Professor Arthur William Galston during 1981 to 83. He is a recipient of Biotechnology Overseas Research Associateship from Department of Biotechnology, Government of India, and worked as a visiting scientist at University of Minnesota, St. Paul, USA, in the plant genomics during 2001. He has completed research and development projects worth rupees more than 4 crores, sponsored by various funding agencies. Professor Shekhawat has published over 190 research journals in research papers in journals of national and international repute. He has also co-edited 4 books in the field of plant tissue culture, molecular markers, plant breeding, and gene editing. He has more than 3,500 citations, H index of 36, I10 index of 95, and RG score of more than 35. He has taught cell biology, genetics, molecular biology, and plant breeding to undergraduate, postgraduate, and the PhD students. He has been mentor of 43 PhD students, guided a number of MPhil students, postdoctoral fellows, young scientists, including UNESCO fellow. He is presently a member of peer team of National Assessment and Accreditation Council (NAC), and also a member of State Higher Education Council, Government of Rajasthan. He is member of Board of Governors at Indian Institute of Technology, Jodhpur, Rajasthan, and he is also serving presently as a national correspondent of International Association of Plant Biotechnology, India chapter for more than last 10 years. Beside these, he has a number of achievements in his credit, but it will take a very long time to list all of them. So with these few words, I would like to welcome you, sir, in this webinar, and request you to please present your presentation, present your deliberation, present your talk. Over to you, sir.
Thank you, Ashok. I think Professor Shekhawat will be joining now with his lecture. Welcome, sir.
I greet all the participants of this webinar. I'm Dr. Shekhawat. I'm thankful to the Vice Chancellor of Narain Vyas University, Jodhpur, and the Head Department of Botany, Narain Vyas University, Jodhpur, for supporting organization of this webinar on recent advances in biotechnology. I joined the Department of Botany in 1974. I was born in 1953 when Watson and Crick's model of DNA was reported, and I will try to tell you about the recent advances in biotechnology, because I have been part of development of biotechnology, plant biotechnology. I have been associated with the organization of laboratories for biotechnology. Luckily, I have been to various institutions with various scientists at Yale University, University of Minnesota, which have been participating in development of tools and technology of molecular biology and biotechnology. So I will share those experiences with you. Next slide. Well, there was time when biology and zoological sciences or botany were not considered very important subjects. And Ernest Rutherford gave a statement that in science there is only physics, all the rest are stamp collections. It was an unfortunate statement, and he got Nobel Prize in chemistry. But if you see the progress in the last 200 years, you will see that next slide. We have entered in 21st century, and this century is century of biology. Carl Booz wrote review article in on new biology for new century. Then there is a book published by National Academy of Sciences 2009. Everybody can read this book because this is freely available. It says about the modern biology for 21st century, where there is combination of biology, tools of molecular biology, computer, bioinformatics, and number of things. It is basically book to suggest interdisciplinary approach for understanding biology to solve the problems of human beings. There are many other books which are available on from National Academy of Sciences. One can approach and get the copies of these books, very important books. Next slide.
Sir, I would interrupt you just for a second, sir. We are not able to see your presentation. I'll ask Ashok to correct that thing. Ashok, we are not able to see the presentation. Can you start presenting and then sir will start? So you are visible but not your presentation, sir.
Is it visible now?
Now it is visible, sir. Please continue.
Yes, it is visible.
Yes, is it visible? Yes, now it is visible.
Can you see slides now?
Yes, sir. Yes, sir. Now we can see. Sir, please continue. Sorry for interruption.
Okay. So, there is a book written by Thomas Friedman. Friedman is a very good internationally known author writing books on various subjects. And the title of book is Thank You for Being Late. And this is book on the optimistic thinking of age of accelerations. And so, and in this book, there's mention on page 144 of IIT Jodhpur and Prem Kalra Saab for contributing in the development of technology for armed public. We are surviving under conditions where things are under acceleration. And if I remember, in 1981 there was simple computer. In 1985 it was 186, then came 286, then came 386, then came 486, then Pentium computer, and there is an acceleration of information technology. And then there are accelerations in several other areas also. This slide shows the cost of decline in the cost of sequencing of genomes of human beings. And in the 2000, the cost was too much high. It was not in. Nobody was thinking of sequencing human genome, but in 2000 now you have genome sequencing with $1,000, and anyone can go for sequencing of genome. So speed and the capacity to store information are accelerated. Next slide. Well, there are many types of accelerations. One acceleration is rising population, and you can imagine China has 1.4 billion population, India has 1.38 billion population, US has 331 million population, and there are a number of other countries. I have enlisted 11 countries which are contributing in population rise. This population rise has caused accelerated degradation of resources and accelerated use of energy, accelerated use of instruments and equipment. Next slide. And in this acceleration, you can also see the rise in carbon dioxide concentration, which is now 416 ppm, very dangerous, causing climate change and global warming. Next slide. Well, we are living in the period which is now called as Anthropocene, which is part of Holocene, and the Anthropocene is characterized where everything is being controlled by human activities, and this is characterized by 4 or 5 characteristics. First is rising population, which is with the accelerating rate, rising pollutions, all types of pollution, chemical pollution, water pollution, and the situation is so bad that in the areas where we are producing food, soil and water is polluted, and that is coming in our drinking water resources. We should know about our Punjab, where highest yield was achieved in wheat production during Green Revolution, and there is pollution of soil, and there are cancer patients coming from Punjab to Ganganagar, Bikaner, because of overuse of resources polluted. Then we have accelerated use of plastics, and people say that Mother Earth is now wrapped in plastic, and plastic is dangerous now. It has polluted our river Ganga, Yangtze, and Yellow River of China. A very bad situation. Then we have plundering of biodiversity and environment. You know, human being rising population has killed population of large number of plant species, animals, and microbes. I will give you 2 examples. In Rajasthan, when I was child, the entire sandy area was covered with Caligonum polygonales, and there was time when we used to cut thousands of plants for cleaning the fields for agriculture, and now there is not a single folk plant in the desert sand dune sticks, which is just like a sort of tsunami causing in the biodiversity. The other plant which disappeared during this period is Comifora whitei, the Google plant. Before 20 years, we had in a village called as Balarwa near Jodhpur, forest of Comifora whitei, and the forests have disappeared. So this blunting of biodiversity because of rising population is very dangerous, and it is very difficult now to control it, and if you don't control it, it will cause damage. There are around 7.7 or 8.5 or 8.8 million species, which comes around 84 lakhs, lakh chaurasi of our thinking. We don't know exactly how many species are there, but these species are very important. Then you have rising poverty because of population or overuse of resources. Then tendency of profit making. You know, the multinational companies are causing capitalocene. Then everywhere there is preparation of war. So the Anthropocene is a situation where these things are accelerated, and we are reaching tipping points where anything can happen, like the mega fires in United States and mega fires in Australia, and floods and heavy rainfalls and damages in mountains. Next slide. Well, these are 4 types of forces on Mother Nature, which are described as quadruple speed human growth population. 80% of them are poor, 20% are very rich, and they are 20% are using 80% resources, 80% using 20% resources. Then we have force of rising carbon dioxide concentration, greenhouse gases, which is now reaching around 410, and other gases, methane and other gases which cause global warming are also increasing. And then we have decline of ecosystem services. Most of the ecosystem services are disturbed and destroyed, 60% lost, and it's a very warning type of situation. And under these circumstances, there are chances of surprise dilemmas which we are seeing now throughout the world. Next slide. Well, there are a number of thinkers and writers and scientists, ecologists who are worried about these accelerated usage of natural resources. There is a book by E.O. Wilson, published in 2016. This book is entitled as Half Earth. In this book, Dr. Wilson has argued that using biotechnology and other technological advances, we can spare 50% of Mother Earth for conservation of biotechnology, conservation of bioresources, and conservation of plants and other species. In this book, they have identified 35 good places in the world, in the biosphere, and you'll be very happy that out of 35 or 36 good places in the biosphere, one is Western Ghat, the other is our Sikkim area, and I suggest that many of us should read this book. This is a beautiful book. This says that living beings or human beings require oxygen, in this book, it has been suggested that if all animals disappear from the planet biosphere, nothing will happen. But if plants disappear, everything will happen. And this book is basically for praising and supporting survival of plant species and forests for well-being of human being. Next slide. Well, then there are situations where people are realizing that there is limited interest of botanists or scientists in plant sciences, and in the recent issue of Trends in Plant Science, the paper published, title is The End of Botany. It suggests biology is unable to recognize common plants, so we have lack of taxonomists, decline in botany students, faculty and courses, university departments, herbaria, highlighting the current erosion of botany and interest. And therefore, it's very alarming. How did we reach this crisis, knowing that plants form basis of life? What are the causes and what can we do to reverse this? This is a subject for our consideration. During this period, Tom Crother of Yale University, he was postdoc there. He surveyed forests of the world and calculated number of trees, which was found to be 3.04 trillion. And it was calculated that in average there are 422 trees for each human being on the earth. And if you compare that situation with the Indian scenario, you find that we have only 27 to 28 trees per person, probably lowest except the Middle East countries, desert countries. Brazil has 1494, Canada has 8,900 species, China has 102, 3 times or 4 times more than India. So the scenario in India is very poor. We should save our trees, we should promote propagation of trees, and we should realize that trees are systems which support life of animals and plants, control climate, bring monsoon and rains. Next slide. There are 2 journals, very important journal recently introduced. One is Plants, People and Planet. I suggest that young scientists should read this journal because this brings beautiful reviews on application and importance of plants. Then there is another review with journal which is Plant Direct, published by Vyas. I feel that I read regularly these issues, and these have beautiful reviews and papers on importance of plants and application of different types of technologies for amelioration of the plant systems. Next slide. Well, for Indian students, and this is a photograph of Dr. Govindji, and there's no surname and first name for Govindji, so he has now started Govindji. He's world-known expert on photosynthesis, which produce biomass of 70 multiplied by 10 raised to power 13 kilograms per year, and plants are therefore major source of biomass production. Uh, the plants produce biomass, and this is second largest event on Earth after rainfall. So, photosynthesis research is very important, and there are a number of people who are trying to understand biology of photosynthesis and application of biotechnology in promoting productivity of plants. Next slide. Yes, I must tell you that Melvin Kelvin, who discovered the carbon fixation cycle, was product of University of Minnesota, undergraduate from University of Minnesota and postgraduate from University of Minnesota. Luckily, I was at University of Minnesota as visiting scientist during 2000-2001, and I can imagine that there are a number of scientists who have contributed in production of biomass through photosynthesis. Next slide. Well, there is another review article which all of us should read, and this is written by Dr. Peter Graysoff, who is biotechnologist, originally in tissue cultures of legumes. He published paper. The title of the paper is "All We Are Saying Is Give Plants a Chance." He felt that there is no Nobel Prize in plants or botany or plant sciences, therefore there is limited popularity among press, and therefore importance of plants and botany is limited. So sadly, there is no Nobel Prize for plant sciences, preventing the popular press from featuring achievements in this area. We should remember that Norman Borlaug worked in plant pathology, improvement of wheat. So the entire thing was botany, but he got Nobel Prize in peace because there was no Nobel Prize in botany. Still, there is no Nobel Prize in plant sciences. Norman Borlaug was again product of Department of Plant Pathology, University of Minnesota, where Dr. Stickman was plant pathologist. He supervised him, and both of them got involved in Rockefeller Foundation support for promotion of green revolution, which came from Mexico to India to Pakistan. I hope all of you know that Swaminathan Saab was a person who promoted green revolution in India through Norman Borlaug. Then another person, Dr. Barbara McClintock, she studied corn, maize, and transposons and genetics of maize, and got Nobel Prize in medicine because again there is no Nobel Prize in plant sciences. So it's a very bad situation so far as press and popularity is concerned. But still, plant sciences, scientists and plant sciences are important because plants fix carbon dioxide, which is toxic, and therefore plants are living Nilkanths or Sakshat Shiva. You can imagine that producing oxygen using carbon dioxide. Plants create no pollution. They do not release unwanted material, fecal materials, and they do not urinate to pollute the environment. Then there are very interesting things about plants. Plant cells are totipotent or pluripotent. As we know that pluripotency and totipotency was expressed in plants in culture. Then Mendel's laws were discovered on peas, not on beets, in 1865. Cloning of differentiated somatic cells was in tobacco. People have in the Dolly very popular, but Dolly is not true cloning. It was tobacco true cloning, which was done in 1965 by Bimla Basil and her mentor. Then you have number of other things which are very specific to plants. Say example, gaseous hormone discovered was not nitrous oxide but first three. Then first morphogen, growth regulator which controls the morphogenesis was not retinoic acid, but it was cytokinin. And the tissue culturists know that balance of cytokinins and genes is important for inducing morphogenesis, getting regeneration. Then catalytic RNA ribozyme was discovered in avocado, and we must know that Sidney Altman and Thomas Sheck got Nobel Prize in ribozymes for ribozyme discovery, and this was catalytic RNA. The transposable elements were discovered in plants, not in yeast or bacteria or fruit flies. It was discovered by Barbara McClintock, so plant biology is very important. Then last very interesting thing is that there are three RNA polymerases in eukaryotes, RNA polymerase first, second, and third. But in plants, you have RNA polymerase 4 and you have RNA polymerase 5, which are involved in gene silencing. Next slide. Well, Department of Botany, Narain Vyas University, we started use of biotechnology right from 76. Professor H.C. Arya, Harishchandra Arya sir was our head of the department. He was our mentor. I suggest that he was man ahead of time because he thought of applying biotechnology, developing biotechnology laboratories in the Jodhpur. And because of his wisdom, large number of people were trained at Jodhpur, and we established laboratory in 1974-75. There was no laminar flow bench. Now we have, I think, more than a dozen laminar flow bench in the department. There were no flasks. There was limit of sugar in the laboratory for making media. And now we have plenty of flask bottles and culture bottles for large-scale propagation of plants, and internationally known laboratory for micropropagation of trees and plants. And we have very well established greenhouse. This was because of Professor Arya and his wisdom. But then this lady, Radha Rani Arya, Madam Mrs. Arya, she was silent worker for helping the scientists and science. She worked from home, and I always feel that she was force behind all of the scientists who developed, who were promoted by Professor H. Arya. Next slide. Now, then you have the applications of biotechnology in various fields. You know, original maize was domesticated from teosinte, and the seed of teosinte are not even edible and not liked by anyone, but human being applied scientific principles and plant breeding methods and developed maize, which is now different types of maize, corn flakes, baby corn, giant corn, sweet corn, and you have popcorn, types of corns. And this is application of scientific principle in improvement of productivity of maize. This is heterosis, discovered at Connecticut Experimental Station in 1907-8. These are crops of hybrid maize through domestication, and these are crops of maize with the effect of transposons on seed formation. Next slide. Well, then the domestication was also done in tomato. The original tomato was very simple, but then it was bred through principles of genetics, through plant breeding, and now we have tomatoes which are of different size, with different colors and different tastes. So we have been doing domestication, super domestication, and now application of biotechnology. So we have de novo domestication, which are now described as 1G domestication, 2G domestication, 3G domestication, and now the 4G domestication, where the designer breeding is being done. Next slide. This example of rice was improved through hybridization. Dwarf genes were introduced in rice to improve the rice quality and productivity. This is super rice, and then genetic engineering was applied for production of golden rice, first golden stew, which was dream of Dr. Ingo Pottikas, a Swiss scientist. Next slide. Then there are scientists in the US and other places who are trying to think of application of artificial leaf for harvesting of sun. And this is one example done by research done by Dr. Nocera at Harvard University, where artificial leaf is used for production of biomass, which is utilized by bacteria, and bacteria utilized for promoting growth of radish. So it's a beautiful example of application of technology for increasing productivity. Next. Well, since biotechnology involves basically cell or tissue culture, this slide I have incorporated in order to compare animal cell culture and plant cell culture. As I said, animal cell culture, animals are important because human being is involved. Plants are attracting less attention and therefore limited resources for plant biotechnology, but still plant biotechnology was started in 1902 by Haberland, Gottlieb Haberland, who attempted tissue culture. Then Cote and Robbins in 1922-20 established root culture. Then P.R. White established continuous culture of tomato roots. So basic tissue culture was established. Then you have application of auxins and cytokinins in culture. 1939 by P.R. White, Godfrey Novikov, cytokinins applied in the medium. So you have by 1945 you have defined medium which was called as White's medium. And along with plant cell culture in 1905, Rose Harrison established cell culture at Yale University. I visited the place where the culture was started. And after that, cell culture in animal systems has been used for a number of purposes, including gene transfer, genetic engineering of mouse, and number of other systems, which we will discuss later on. So, next slide. So, well, when it comes to plant tissue culture, we have number of persons who have contributed. This is P.R. White. He loved India. He came several times to India and passed away in India, Pune. He was one person who established long-term culture of tomato and very important contribution in designing medium, defining conditions of culture. Then Robbins in 1922 established root cultures. This paper was published in Botanical Gazette, which is now New Phytologist. Then this photograph is of Professor E.C. Cooking, who was pioneer in isolation and culture of protoplasts. He isolated protoplasts from tobacco, using enzymes produced by pathogens. And we should know that Dr. Cooking has trained large number of Indian scientists. Dr. S.S. Bhojwani, who wrote a book, beautiful book. Dr. Deepak Pental, very well known geneticist and former Vice Chancellor of Delhi University. S.L. Kothari Sahib at Jodhpur. And therefore, area of plant protoplast culture, protoplast fusion was identified and applied. Next slide. In this line, we have all of us know about Murasike and Skoog's medium, which was defined in 1962. But this is Skoog, this is Murasike, and the third person, T. Thor. It's very interesting that Thor did PhD in Allahabad, in India, and he liked Indian culture, including papads, and then he became professor at the University of Calgary, where he established Tissue Culture Center, which is recognized now as United Nations of Plant Tissue Culture. He passed away last year. He trained large number of Indian scientists, including Kamlesh Patel, Kiran Sharma, who is now at ICRISAT, and this lady Indra Harry is settled in US. And there are a number of other scientists. I got fellowship to work with Thorpe, but I could not go there. I had links with him. A great scientist who defined the conditions for tissue culture of trees, particularly gymnosperms, Pinus cycas. We said one paper, publication of one paper with Thorpe, Kamlesh Patel, and Graham Burley. It was on culture, isolation and culture of Pinus species. So we, US and India and Canada were involved in publication of this paper. Next slide. Well, then there are many Indian scientists. If you survey the development of biotechnology, you will find that large number of Indians settled in Canada, United States, Germany, UK were trained in India. And Professor Panchanan Maheshwari of Botany Department, Delhi University, promoted such school, which produced Drs. S.C. Maheshwari, Dr. Nirmala Maheshwari, Dr. Sudhir Sapori, Dr. H.Y. Mohan, who was expert in bamboo tissue culture, Dr. S.S. Bhojwani, who is known in tissue culture, large number of other scientists who were trained, large number of scientists who were trained in other countries. In 1971, we organized symposium on plant morphogenesis, where large number of internationally known scientists came to Delhi. This is photograph of Dr. Sipra Mukherjee, who worked with Dr. Satish Maheshwari on culture of anthers and developed technology for haploid culture, which has applied values. Although it is not very well recognized in India, but internationally there are several reviews and books on haploid culture. If there was Nobel Prize in plants, probably this would have gone to Dr. Sipra Mukherjee and Maheshwari Saab for development of haploid culture technology. This is photograph of Dr. Sunil Purohit, Professor of Botany at Udaipur, Dr. Rama Utsap, Professor of Botany at Udaipur, Dr. Sudhir Sapori, former Vice Chancellor of JNU and student of Professor Maheshwari, and very important biotechnologist. Okay, next slide. Well, then the tissue culture has many challenges. This is photograph showing explant collection from trees, mature trees, and the season and location of explant is very important. This is something, it's a sequoia tree. You know, sequoia trees are 150 meters tall. And if you want to establish culture from sequoias, you have to go on top of the plant to collect explants. So this is some gentleman collecting explants of sequoia. Very dangerous. Next slide. But still they collect explants, process them, sterilize them, and raise plants through culture. Next slide. The technology of plant tissue culture has very strange applications, and one of the outcome of the technology is that regeneration of whole fertile plants from 30,000 year old through tissues buried in Siberian permafrost. So the tissues were frozen for 30,000 years. These were retrieved, established in culture, and then plants of silane were raised. So this is a beautiful example of application of tissue culture for retrieval of biological material, conservation of biological material. Next slide. Under these hundreds and thousands of bottles of Johnny Walker being used for multiplication of orchids in Thailand, this has been established as industry, home industry in Thailand for propagation of orchids, and therefore beautiful example of application of biotechnology for business. Next slide. Sometimes, the discipline and commitment and timing is important for maintaining tissue culture laboratories, for hardening of plants, transferring these to the field. So Thai government decided to establish laboratories under control of defense forces. This is laboratory where the forces are responsible for establishment of culture, propagation of culture, hardening in greenhouse, and then transfer to the field. Next slide. This was taken in 1996 in Bangkok. Next slide. Then at Jodhpur, with blessing of Professor Arya, we established tissue culture laboratory, micro propagation facilities, and worked on large number of plant species for multiplication. This is an example of cloning of selected plant of citrus which produces fruits 3 times a year. And these are rooted fruits, and then field transfer plants produce fruits. So this is a beautiful example of cloning of citrus. The technology was taken by my friend Kisan Bhatt at Baroda. He multiplied plants and used the technology for propagation. Next slide. This is another example of application of plant tissue culture. This is in collaboration with Dr. Sunil Purohit of Udaipur, where chlorophytum borevillianum were multiplied in culture. These were rooted and hardened in greenhouse. This is situation in our Jodhpur nursery. Next slide. Well, then in forestry, there are applications of plant tissue culture. This is an example of Silastus pediculatus, Malkangni, very valuable plant. We got one plant from forest department and raised 5,000 plants, transferred them to the field. So beautiful example of application of soft biotechnology for conservation of germplasm and propagation of valuable species. Next slide. Then there are other systems where we worked at Jodhpur. This is an example of cloning of Caparis decidua. We established cultures from mature plants. Cultures are being multiplied in bottles. Then these are rooted and hardened and transplanted to the field. They produce flowers and fruit. This is another species of Caparis spinosa, multiplied by Dr. Vinod Kataria at the department. Wonderful achievement because large number of fruits are producing plants. Next slide. These are clone plants of Caparis decidua transferred in field in the new campus of Jodhpur University. I think beautiful example of application of technology by traditional universities with the help of DBT and EST. We established lab and greenhouse facilities. Next slide. Another 3 species, Metinus emerginata, the Harikankari, which was cloned in the establishment of S-plant, multiplication of cultures, hardening of tissue culture based plants, and field transfer. It's not very easy to multiply trees. Jodhpur is one center where large number of plant species, including eagle marmelos and number of other Salvadora, persicans, these were multiplied through culture. Next slide. One beautiful example of application of biotechnology in forestry is application of tissue culture in Enogaceous species. There are 4 or 5 species of Enogaceous which used to cover the forests of the Aravallis. There is no propagation. In fact, there are no planting materials for Enogaceous in the nurseries of forest department except our nursery. And we established cultures of these 4 or 5 Enogaceous species right from 85, supported by Department of Environment and Wildlife, and established technology. These are cultures of Enogaceous pendula, and Enogasis sericea, Enogasis letifolia, Enogasis acuminata. This is greenhouse where the plants are hardened after rooting and plants are loaded in truck for transfer to the field. There is plant raised through tissue cultures standing in the Department of Botany. The plants were transferred to various places with the help of Forest Department. Then the Tata Energy Research Institute came in picture. They established greenhouse at Delhi, hired two of my students and produced hundred thousands of plants of Enogasis pendula and letifolia. Protocol was developed by us, but it was never acknowledged. This happens in this country, and practically it was without acknowledgement. Next slide. Then you have other plant species which are rare and threatened. This is case of Ducadi erythraeum, which is a seasonal plant, and it leaves, it kills hunger and thirst, and therefore very important but threatened. Limited plants are available. We establish cultures of this system, and this regenerates through somatic embryogenesis. Beautiful example of application of plant tissue culture and regeneration of plants through somatic embryogenesis. Next slide. This our culture facilities where thousands of plants are simplified, multiplied. These are rooted and hardened in greenhouse. This is Carallum idolis, the pempa of Pakistan and India. Very important plant for as vegetable and as medicinal uses. Next slide. Well, another plant Glossonema varians, which is called as Khiroli in Jaisalmer and Bardmer district. We established the cultures, multiplied them. It's very difficult to root them, and I hope the technology will be useful for large-scale propagation and conservation of this species. Next slide.
I think we have some electricity problem in an area, and now sir will be presenting with his laptop now. He was on his system, just a 2 minutes delay. Sir, can we take one or two questions, because there are many people who are asking many questions?
Oh, sure. I will. Sir, question session.
Sure.
There are some students who are asking how micropropagation offers wider opportunity in future biotechnology field, sir.
What is question?
How micropropagation offers wider opportunity in future biotechnology field.
Okay, that is very good question, but I think you have to apply micropropagation technology for specific plant species. I am coming to next slide where we applied micropropagation for large scale propagation of Leptidemia reticulata. Can I reply later on?
Yes, we can continue with the lecture, sir, now.
Okay.
Okay, sir, you can present.
The Leptidemia reticulata jibanthi valuable plant described in our literature. It is used for promotion of lactation in animals and also has application in poultry industry. We established tissue culture at Jodhpur on advice of some experts in Ayurveda and established protocols and transferred the plants in the field. Dr. Vinod Kataria did work on micropropagation of Leptidemia reticulata. This is field of reticulata plants raised through tissue culture. Next slide. चलेगा ना? चलेगा. Yeah, the products are harvested, processed, and marketed. So it has great market, and one can think of application of silviculture in such type of plants where there is market demand and these plants can be propagated through tissue culture. Next slide. And there are other medicinally important plants like Arnibia spidissima. You know, there are, it's beyond thinking that this can be used for industrial production, but you can establish cultures of Arnibia spidissima. This can produce dye in culture. This is an example of in vitro production of ciconine and alkanines, and we must know that ciconine was produced by Japanese in large scale, and this can be one area where some people should invest some money or government should support for production of these dyes in culture, because these are important for treatment of number of like burning cases or number of microbial infections. Next slide. Then we developed, besides tissue culture, at Jodhpur we developed tools of aeroponics and hydroponics. I think this will address to the questions of people. And this project was supported by Department of Science and Technology, Government of India. This is an aeroponic system designed for large-scale rooting of difficult to root plant systems, including Leptadenia, Tinospora, and number of other systems. Okay. So this is a beautiful example of application of technology for large-scale production of difficult to root systems, including trees. Ashok, next. And these are rooted plants of Leptadenia reticulata through aeroponics, and this can be done in other species also, including trees of several systems. Next slide. Ephedra foliata, a very important plant, endangered and threatened. In the climatic conditions of Rajasthan, this has application in health. It's called as oat fog. The other ephedra, the upper slide is Ephedra girardiana, which is in Leh and Ladakh. Unfortunately, this is smuggled out as food. Next slide. In the desert area in our country, we have large number of leguminous species which synthesize fixed nitrogen using different types of microbes in the roots. This is photograph of legumes of Indian desert. The research is being done by Dr. Hukum Singh Gehlot and their team for promotion of nodulation in the legumes for providing fertilizers in our soils. Next slide. Then the root research is very important, particularly when there is stress of salts, stress of water, and when there are situations where roots are responsible for survival of the system. And the subject is now called as Savelomics, and this is one part of biotechnology in Australia, United States, where root phenotyping is being done, and root biology is used for promotion as new green revolution for increasing productivity of plants. Next slide, particularly crop plants. We have large number of plant species in our country. This is beautiful example of a plant, Caligonum polygonadis, the humble service provider in the threatened area. I mentioned that this plant was covering the entire sandy areas, now has been removed. This plant is providing fuel and fodder wherever it is available. It was used for fodder and fuel. These are beautiful flowers of the plant. It has it's very soft plant material. Next slide. And it can be propagated through cutting of stems. This is fodder stored in Jaisalmer from Caligonum. This is fodder for camel fruits, which are called as Gantial. There is a village, Gantiali village, after this name of this plant. Then there is Gantiali Mata in Jaisalmer. The other product in between this is buds of Caligonum polygonidis, which are loaded with antioxidants. These are mixed with yogurt or charge for treatment of heat strokes and therefore stress remover. Next slide. This is greenhouse established at Jodhpur, Department of Botany, with the help of DBT, and this is one greenhouse which is established in traditional universities and working for last 25 years. It has been used for hardening of tissue culture raised plants of, I think, around more than 25 species. Beautiful asset and example of application of technology for amelioration of biological resources. Next slide. Well, then at Jodhpur we use soft biotechnology, and I have given examples where these were applied for desert plants. If you analyze biotechnology, biotechnology is getting services and products from living organisms and systems. The term was given in 1919 by Hungarian engineer. If you analyze the history of biotechnology, you will find that it can be divided into three categories. Ancient biotechnology, where our forefathers applied technology for making wines, yogurt, and bread. Then you have classical biotechnology, which we are using for last 200 years. This is application of tools and techniques of plant physiology, plant growth regulators, hybridization technologies, plant breeding for improvement of crops. Then you have modern biotechnology, which started from 1945 onward, with the development of molecular biology, with the development of cell and animal tissue culture, with the development of new tools and techniques for genetic engineering of cells. You have introduced number of products, including golden rice, and therefore, next slide. Well, I have two examples which can be classified as which are used in ancient biotechnology and which are part of the modern biotechnology. Yogurt or dahi is one example. Everybody knows that the starter is used by mothers for processing yogurt. But then we should realize that yogurt technology is part of food technology, and the latest CRISPR technology is being used for improvement of yogurt production. In fact, CRISPR was originated through studies of yogurt in Europe and America. Then another example of ancient technology or technological resource is ability of waters of Jamuna and Ganga for cleaning the water. These waters have ability to kill bacteria in the system, and they, in 1896, Henkin published a paper in which it was analyzed that the waters of Ganga and Jamuna have ability to clean water and kill bacteria. It was, the term bacteriophage was not known at that time, but the action of bacteriophage was discovered in waters of Jamuna and Ganga, and I feel that this characteristic of our river should be understood and studied, and bacteriophages phages should be multiplied by application of any type of technology. Russians are overproducing bacteriophages for treatment of number of diseases and cure of number of problems. So we can use some technology for large scale production of bacteriophages, and probably can use these for Ganga and Jamuna water cleaning. Next slide. While all types of biotechnologies are dependent on some basic principles, and in 1838-39, scientists studied the biological systems and gave one unified concept, and that is called as cell theory. It has 4 components. The theory was given by Slyden and Swan. Slyden was a botanist and Swan was a zoologist. They were friends, studied independently. They concluded that all living organisms are either unicellular or multicellular, prokaryotes or eukaryotes, are composed of cell or cell products. It's very important point. The second point is that chemical reactions, including energy generating pathways and ATP synthesis, do occur in single cell or cells. The third component is cells arise from pre-existing cells, that means mitosis and meiosis and continuity of life. Then fourth important component, which is basis of modern biotechnology, is that cells contain the hereditary information or genetic substances of organism of which cell or cells are part, and this information, basically DNA, is passed from parent cell to daughter cells, and this is basis for the molecular biological understanding of physical, biological and chemical nature of genetic material, and this was later on used for development of modern biotechnology. Next slide. Well, last year in 2019 we celebrated 150 years of number of things. The periodic table was discovered in 1869 by Mendeleev. Mahatma Gandhi was born in 1869. Then nucleic acid were isolated by Miser and characterized in 1869. The journal Nature started in 1869. So these were events in 1869 which are responsible for promotion of biotechnology and promotion of democracies in the world. In the same period, in 1859, Charles Darwin published Origin of Species. Louis Pasteur rejected the concept of spontaneous generation, and then Mendel discovered Mendel's law of inheritance in 1865-64. Next slide. Then, the Mendel's law and cell theory originally were considered basic, but now we know that these were challenged. The cell theory was challenged because some components of cell theory are not applicable for viruses. Some components of cell theory are not applicable for blood cells. Similarly, the Mendel's law of inheritance is also challenged because of findings of new concepts and new biology in systems. Say example, plastid contains genetic information, mitochondria contain genetic information, and these have uniparental inheritance and do not follow Mendel's law. The transposons came in picture which do not follow Mendel's law. Then you have, for last 30 years, RNA editing in plastids and mitochondrial systems which do not follow Mendel's law. Then you have concept of chromothripsis, sudden disruption in the organization of chromosomes, very important now in the era of CRISPR. Then we have RNA interference through long non-coding RNAs or circular RNAs or small RNA or microRNA, which are responsible for gene silencing. Then you have chromatin remodeling, histone modification, and chromosome engineering being done in Mother Nature. So you have concept of epigenomics and epigenome editing, genome imprinting, and latest is now gene driving and gene editing, which is through CRISPR and CRISPR technology. I must tell you that right from 1869, there were several types of studies and developments which characterized the hereditary materials in 3 lines. One line was characterization for biological characteristics of hereditary material. Second was chemistry of hereditary material. Third was physics or physical organization of hereditary material. So you have chromosome theory of inheritance, which was challenged by Morgan and later on supported by Morgan. Then you have in 1902 Haberland establishing tissue culture technology for propagation of cells in culture. Archibald Garrett established the concept of genes responsible for disorders. Then you have a concept of genetics and concept of genome given by botanists. And Morgan ultimately told us that the chromosomes are carriers of hereditary materials which are organized on chromosomes, linearly arranged on chromosomes. And Fulgen established technology for staining of chromosomes and observing these chromosomes under microscope. Then in 1916-17, Levny told us that the DNA is made up of tetranucleotides, which was not found by true later on. Then you have Griffith and his observations and his studies in 1926-27, telling us that there is something transmitted from one generation to another generation, from infective bacteria to ineffective bacteria, and the transforming factor is responsible for number of characteristics of organisms. Oswald Avery and MacLeod and McCarthy in 1944 told us experimentally that hereditary material is DNA. The transforming factor was DNA. Large number of people opposed it also, and Avery did not get Nobel Prize because up to his death, the people said that this is not true. When this was done at that time, some people, when it was proved that genetic material is DNA, then the physics of genetic material was studied, chemistry was explained, and it was Chargaff who told that the ratio of GC and AD. Then Linus Pauling told us about the double hydrogen bond and triple hydrogen bond between A and T and G and C. And then Alexander Todd told us that there is phosphodiester bond in nucleic acid. Then it's very interesting that Corona, who discovered later on genetic code, he studied with Todd, Alexander Todd, and Linus Pauling got 2 Nobel Prizes. One was for discovery of chemical bonds, the other was for peace. In 1953, this Watson-Crick model was given. Crick explained central dogma of biological sciences or molecular biology. Hamilton Smith discovered endonucleases, restriction enzymes. Therefore, from 1869 to 1965, we have 3 or 4 lines of developments which promoted biotechnology. Next slide. You know, the tissue culture became very effective. MS medium was defined. Tools and techniques of aseptic conditions were defined. Culture conditions were defined, and in 1961, protoplast culture technology was developed. So you have plant material which can be treated with enzymes, and the cells are converted into protoplast. Then you have protoplasts of Vigna, which are around 50 micrometers. These protoplasts can be cultured, amended, these can be fused to produce somatic hybrids. And it was Peter Carlson in 1972 who fused the protoplasts of two species of tobacco and produced somatic hybrids. Later on in 1978, hybrids of tomato and potato were produced through fusion of protoplasts. Next slide. Then, each cell has nucleus has genetic material and chromosomes. And then plant cells also have mitochondria and chloroplast, which are carrying genetic important information, plastid particularly for photosynthesis, Rubisco system. So people thought of amending the protoplast, amending chloroplast, amending mitochondria. Next slide. This is a protoplast being micro-injected under microscope for gene transfer and fluorescence dyes. This is protoplast of animal systems micro-injected under vacuum. So, मैं change करूँ क्या? Next slide next slide. Well, the protoplast fusion technology was used for production of somatic hybrids, and this is an original photograph of hybrid between tomato and potato in 1978. So potato being produced and tomato produced, so tomato-potato hybrids. Then this protoplast culture technology was also used for production of hybrids where one system was enucleated and therefore only cytoplasm or mitochondria and plastid were fused with another protoplast. So hybrids were produced in large number of plant species, including citrus and brassica species, new area of research. Next. During this period, it was realized that crown gall is produced in plants, and crown gall was studied right from 1939 onward in order to understand cancer. And it was found that crown gall is caused by Agrobacterium tumefaciens, and for continuous growth of the crown gall, presence of Agrobacterium was required. But later on, it was realized that if you cultivate crown gall cells, these are autonomous for oncogenes and cytokinins. And later on, Morel discovered that the crown gall cells have genetic information, chemicals, agropine and nopalines, which are produced by bacteria and now produced in plant systems. So indication of gene being transferred from bacteria to plants, and therefore these chemicals were used as markers for transformation. But then later on it was realized in 1972 or 1 in Belgium that genetic information from bacteria is transferred permanently and integrated with plant cells. So plasmid was discovered, and plasmid was called as TI plasmid, which was 200 kilobase. Later on, this plasmid was amended, disarmed, so to make it safe, and then it was made binary plasmid, then it was made super binary plasmid, and then it was used for transformation of large number of plant species, trees, cereals. And by 1995, it became possible to transform plant cells using Agrobacterium tumefaciens plasmid, and large number of products were produced right from 1983 to 2000. Large number of plant species, GM crops were marketed also. Next slide. This is an example of genetic manipulation in roses. You have beautiful colored roses and blue rose. Next slide. Well, then suddenly people realize that if you do transformation not of nucleus but of plastid, it is easier to to do the transformation for some specific objectives or production of some foreign proteins in plant cells. And Henry Daniel, scientist of Indian origin, perfected the technology of transformation of plastids. So plastomic plants were produced. Henry Daniel became very famous in the United States. He's now working for hospitals for production of foreign proteins in plant systems. He's now at University of Pennsylvania. These are photographs of Henry Daniels, who is also editor-in-chief of Plant Biotechnology Journal, a leader, and recently he has published his biography. Next slide. Well, then the genetically engineered crops became very popular in the United States, China, and other countries. Basically, genetically engineered crops for insect resistance and for herbicide resistance were produced in maize, soybean, and brassica systems, and GM crops were transferred to field. I think in the United States, most of these crops are GM crops, but in India we have resistance against the introduction of GM crops. This is an example of GM brassica being opposed in India. Next slide. Well, then, along with plant tissue culture, there were development in animal tissue culture, and these animal tissue culture reached in the United States or elsewhere, discovered many technologies for genetic engineering of mouse, yeasts, and ultimately we have a stage where gene targeting is being applied for various purposes. It's a photograph of Mario Capecci, who did pioneering work on cell culture of mouse for gene targeting, and he found that if you introduce gene from outside, it can be inserted in certain areas, but what he suggested was that the foreign gene introduced was clustered at one place, and he felt that it was because of some specific factor, and it was felt that it was because of homologous DNA joining, and it was because of double strand breaks in the DNA, and therefore he suggested that genetic engineering or gene targeting of human cells or mammalian cell will be done only if you introduce double strand breaks in the cells, and therefore technology was promoted. Then this Sajov Stark बंद कर देंगे उसको यहाँ से बंद है इसकी जरूरत नहीं he was one then Joe Stark was mentor of Doudna, who got Nobel Prize recently. So he suggested gene targeting in animal systems, but then Maria Jessin suggested that gene targeting can be promoted in yeast or mammalian cells if you apply enzymes from outside, and the enzyme which can cut the DNA. But she suggested that if you use the DNA and the restriction enzymes of general nature, it can make large number of fragments of DNA, it can cause damages. So she suggested use of meganucleases, and one of the meganucleases was identified was ISCEI, which came from Saccharomyces cerevisiae, and she found that if you use this enzyme and foreign DNA, the cells can be transformed, the genes can be targeted with very high efficiency, and she got around 10% efficiency of gene targeting, which was very good. This meganuclease ISCE has specificity for 18 nucleotides, so it cuts only limited sites, and in fact in mouse there is no site for cutting of DNA because of this factor. she realized that she has to introduce site for this enzyme meganuclease in mouse, and then only the gene targeting was possible in mouse. Next slide. Then people thought of using meganucleates, which should be further amended and therefore hybrid. हो गया तो आगे चले. Okay, so the hybridization of proteins with meganucleases was suggested, and Dr. Chandrasekharan Srinivasan was pioneer. He joined Dana Carroll, and they produced genetically engineered meganucleases with the help of zinc finger proteins, which was used for transformation of large number of animals and plants. Up to 2006 or 9, large number of systems were genetically amended using this combination. Later on, it was realized that plant pathogen Xanthomonas produces some effector proteins which are called as transcription activator like effectors. If you synthesize combination of these factors with the meganuclease, more specificity can be achieved. And by 2009, these were used and applied in plant sciences and animal systems for gene targeting. Suddenly, CRISPR came in picture, and it was realized that CRISPR has more effective abilities to gene target. Therefore, GFN and TEL were overtaken by CRISPR. CRISPR has a very interesting history. It is clustered regularly interspace short palindromic repeats, which was found first in E. coli by Japanese scientists Ishino in 1987 in E. coli, then Francisco Mojica found in 89-93 in Helophyrex bacteria, and later on Mojica in 1995 found that it is present in around 20 species of bacteria. Interestingly, there are repeat sequences in genomes of many systems. There are repeats like simple sequence repeats, which are used as markers in plants and animals. But then this repeat discovered by Mojica was named by Rudd Jensen as CRISPR, has some specificity, and it was realized that this is present in large number of microbes and bacteria, and it was realized that part of these repeats were contributed by viruses and bacteriophages which infected the bacteria in the past. So this is a type of acquired information from viruses and plasmids, and acquiring adaptive immune system for protection against further infection of the agents. In fact, this was discovered in Yersinia also, and number of systems. The papers on the studies of these systems, CRISPR systems, were processed and submitted for publication. These were rejected by Nature, PNAS, Molecular Microbiology, Nucleic Acid. Took several years to publish in 2005 in Molecular Evolution. This gentleman, Mojica, was Spanish, working in the University of Alicante. Very difficult for him to publish the findings because people rejected with the remarks that these are ordinary things which are present in systems. Then a French scientist discovered that these specific sequences are past genetic aggressions present in the systems. So that means if any pathogens or virus or bacteriophage had infected the bacterial cell in the past, part of this was acquired in the genome, and this is used as antibody against the invaders. Then Alexander Bolotin in 2005 speculated that CRISPR conferred immunity, proposing that from CRISPR locus work by antisense RNA inhibition of phased RNA expression. That means obviously if the sequences are acquired from pathogens or invaders like viruses or bacteriophages, these are transcribed and the transcribed RNA is used against the further invasion. So it is basically RNA-based immunity acquired in the bacterial system. And this was found also in lactic acid bacteria, which are used for making yogurt. And it was later on found that this has applications in yogurt production. So there is say that every time you are using yogurt, you are eating CRISPR, because these are acquired by the bacteria. Next slide. Well, then this technology of CRISPR was used for gene targeting, genome editing in number of systems right from 2009. This is genome edited pineapple, genome edited mushroom, which does not brown if you cut it. Next slide. And then in India, genome edited wheat was produced in 2013. Next slide. Well, the CRISPR technology has been used for genome editing, for elimination of certain genes, from editing of certain genes, and the beautiful example of dehorning of cattle. You know, the cattle have horns, and there are genes which are responsible for regeneration of horns, and they edited the genes and produce animals without horns, so dehorning, which has very important application. Then the genome editing technology was also used for production of animals which produce better meat and better products. So you have transgenic cattle and sheep for commercial purposes, and these are genome edited. The paper was published in Transgenic Research. Next slide. You have beautiful examples of genome editing. Pigs are around 40 kilo, but if you produce pigs with 10 kilo, these are easy to handle, not dangerous, and therefore beautiful example of application of genome editing animal production, micro pigs in China. Next slide. Then you have application of genome editing for production of pig virus resistance, which is swine flu resistance, and therefore example of application of CRISPR technology for producing swines which are free from swine flu. Then there are applications of genome editing technology using CRISPR for production of for amendment of large population through gene drive, and we are expecting application of mutation chain reaction in production of population of mosquitoes where females will be eliminated through chromosome engineering. So beautiful example of genome editing for eliminating dangerous animals. In fact, it has applications in ecology, and some ecologists are posing it. So we can expect elimination of mosquitoes which are dangerous, elimination of pathogens which are dangerous through genome editing technology. Next slide. Well, this technology has application in animals. It has applications in various fields, and it is expected that if permitted, design babies will be produced, which is being opposed. Next slide. The persons who develop these technologies became very popular in the political fields, in the White House, in the CIA. It's a photograph of Jennifer Doudna, who has written a book. Next slide. In plant sciences, gene targeting is being promoted by Holger Putzta, German scientist. He has been working right from 1990 onward, and up to 2020 he has produced large number of genome edited plants, including application of CRISPR. These papers came, this is in-planta gene targeting using CRISPR. It is in Plant Journal, then there are publications, Nature Communication, publication in Nature Plants. So genome editing is being applied in amendment of plants. Next slide. These two ladies recently got Nobel Prize, Jennifer Doudna and this lady, Emmanuelle Charpentier, French lady. Both of them collaborated with each other for application of genome editing technology for production of different types of things. And it has become controversial also. There's fight for patent. There are two groups at Harvard University and MIT who claim patent on application of CRISPR technology. This lady, Doudna, did not get patent, but she got Nobel Prize. And there are review articles. Next slide. Review articles to talk about the application of CRISPR technology. So one paper which came in Cell in 2016, the heroes of CRISPR. There is a lot of criticism of this paper because this was published by Eric Lender, and they say that it was he promoted the interests of one group of scientists and ignored the other group of scientists. So there is another article which is called as Unsung Heroes of CRISPR. This came in July 2016. It's very interesting that 17 persons were involved in development of CRISPR technology, and 2 of them got Nobel Prize, and therefore this became very controversial, and it is still debated, and therefore very important development, but controversial. In fact, Dr. Doudna had suggested that CRISPR technology should be carefully used, or it should not be applied in germ lines of human being because it's going to be very dangerous. So this is one example where inventor of the technology is suggesting banning it or regulating it because it has impacts which will affect the society and countries. Next slide. Then we have resources in our country or everywhere in the world where we find that any large number of animals have very specific genes, like elephants have 20 copies of gene P53 which prevents cancer in elephants, so there is no cancer in elephant and mega animals, and the biology of these systems should be understood in order to prevent cancer or amend cancer. Then there is animal or camel which produces antibodies which are called as camelid antibodies, very specific. These can be used for application for future usage. Next slide. In our system, we are very old civilization, very rich in biological sources. We have medical history, and large number of plant systems are used for sources of nutraceuticals and treatment of many types of disorders. This is Dhanvantri, who came on the cover page of Nature in 2016, telling us that there are a number of Indian herbal plants which can be used for treatment of number of disorders, including bhang, your cannabis sativa. Next slide. Cannabis sativa is now being cultivated in various parts of the United States and European countries. I know many laboratories which are working on cannabis in America also, including Pennsylvania and Georgia. Cannabis is supposed to be containing two types of chemicals which are described as anandamides, because these are used for treatment of types of disorders and diseases. Next slide. Well, this photograph I have included for specific purposes. photograph is of Dr. Craig Venter. He is also called as Craig Venter because he developed technologies and tools and techniques which have applications in modern biology and biotechnology. He has written a book. The title of book is Life at the Speed of Light. He is responsible for development of types of technologies. First, he suggested that he will use sequencing or technology of his own for sequencing of Haemophilus. The project was rejected. By the time it was rejected, he completed sequencing. But then when human genome was being sequenced, he said, either you sequence it rapidly or I will sequence it. So he sequenced the human genome with very high speed. Then he established a company with the help of Hamilton Smith, who discovered restriction enzymes. And this company is now sequencing materials of large number of organisms. So you have community genomics, analysis, storage, and then synthesis of these sequences in test tube. So they created microorganism, particularly mycoplasma, artificial cells, through introduction of DNA synthesized outside in test tubes. So you have the sequencing, storage of information. This can be stored in computer. These can be transmitted to other places where gene machines can synthesize DNA, and these can be transferred to cells. And so you have artificial cells, information transferred from one place to another place, and therefore life at the speed of light and teleportation of genetic information. Next slide. Well, this photograph I have incorporated. He's Dr. Arthur William Galston, my mentor at Yale University in USA. He was person who discovered triiodobenzoic acid, TIBA, which was used for promotion of printing in soybean. But if you use in higher concentration, this can cause defoliation. And this property was utilized by Americans in defoliation of trees in Vietnam. So it was technology used for agriculture, technology used in war. And he therefore suggested that scientists should be very careful and should follow ethics and follow the technologies so that it cannot be utilized for bad purposes. And therefore, social responsibility of science. Next slide. Then I have incorporated several books for your discussions and talks, but I think there's not much time. So the first thing that I wish to present is a book which is called as What is Life, written by Schrödinger in 1944. Interesting thing about this book is that this book was read by large number of these molecular biologists and biotechnologists, including Watson and number of others, and they feel that they have expressed that reading this book promoted their feeling about life. This book has calculated size of gene in 1944. This book also says that the definition of life is given in Upanishad. So this is a very interesting book, very small book, and it's very easily available. So all of you should try to read this book. The Double Helix, written by Watson. Watson has written in this introduction of this book that he was inspired by What is Life, written by Schrödinger.
Sir, slide
So there are other books also, but I suggest that for understanding development of biotechnology, we should read this Double Helix, and there is another book which is called as Eighth Day of Creation by Judson. ये क्या हट गई है क्या? Okay, so I feel that I have tried to incorporate the development of history of biotechnology and the tools and techniques utilized by scientists, particularly bioplant biologists, for production of useful products. So this is stage in 2020 where we can expect large number of products from biotechnology, application of biotechnology for conservation of germplasm or bioresources. Thank you so much. I नहीं ना?
Thank you, sir. Thank you very much. And I hope we all have enjoyed. And now Ashok will present. Ashok, please switch off sir's camera also. That camera is on. If Sumitra can off it. Switch it off. For vote of thanks, now it is Ashok.
Okay. I am very much grateful to Professor Shekhawat for his valuable and knowledgeable presentation, loaded with very useful informations, which covered almost all aspects of biotechnology, but biology as a whole, right from ancient times to classical to modern days, tools and techniques such as genome editing and CRISPR-Cas system. I am sure that all of us got enriched with this presentation, and I assure you, sir, that we shall definitely try to work and follow on your suggestions. So now the 2 lectures is over. Now I would like to request Dr. Sumitra Kumar Chaudhary ma'am for the official vote of thanks. Dr. Sumitra Kumar Chaudhary ma'am, over to you.
Sumitra, please unmute yourself first.
Thank you, Ashok. Good morning, Honorable Vice Chancellor, Professor P.C. Trivedi, Head Department of Botany, Professor Anil Vyas, our most valued invited guests, Professor Anna Shekhawat and Dr. Felix Bast, Convener of Program, Dr. Vinod Kataria, Co-Convener, Dr. Meena Barupal, Dr. Ashok Kumar Patel, and our all participants. It's my privilege to have been asked to propose a vote of thanks on the occasion of National Webinar on Recent Advances in Biotechnology. I, Dr. Sumitra Kumar, on behalf of organizing team and my own behalf, extend a very hearty vote of thanks for our Vice Chancellor to be patron. I also express my sincere thanks to our HOD for their logical support and guidance. I must mention our deep senses of appreciation for Professor Anna Shekhawat for their excellent coverage on biotechnology and biotechnologists in the field of biotechnology. I also very grateful to Dr. Felix Bast for their for providing the information about COVID-19 and use of biotechnology against fight against COVID-19. And at last but not least, I express my gratitude to our participants for being with us and to make program so interactive and successful. Thank you all. Thank you so much.
Thank you all. Thank you all, and thanks for joining. We are ending the session here. Sorry for any inconvenience that was caused due to technical glitch. We will be posting all the PPT and the presentation that were here in an edited form in a YouTube link, which will be provided to you. And thanks again. We'll leave the meeting. Sorry we could not had much time for question and answer because we had already exceeded our time. Thank you. Thanks a lot.
Thanks a lot. Bye-bye.